Welcome to our second class on enzymes. In our first video, we discussed what an enzyme is. We also looked at the types of enzymes, and we also discussed some terms which you'll be using as you are writing your essay on enzymes. And we also looked at how enzymes work by looking at the lock and key. So if you haven't watched my part 1 video, the link is in the description. Go watch it before you watch part 2. So before I start, don't forget to subscribe, comment and share if the video was helpful to you. Yeah. So in today's class, we are going to be looking at the characteristics of enzymes. We are going to also look at the factors that affect the rate of enzymes, and finally we are going to discuss how you can name enzymes which are found in living organisms, and finally we are going to discuss the uses of enzyme, which will conclude our topic for today. So to begin, we are going to start by looking at the characteristics of enzymes. So I said that we are going to be looking at the characteristics of enzymes. So what are some of the characteristics of enzymes, or what do we mean when we say characteristics? So characteristics, these are things which differentiate something from the other. So in our case, we are saying that these are the things which will differentiate an enzyme from other substances. So what is the first characteristics? So most enzymes are protein in nature. So that's the first characteristics which differentiates the enzyme, or the first characteristics which enzyme have is that most of them are protein in nature. Protein in nature. That is the first characteristics which you need to understand when you are looking at the enzymes. Then the second thing which you need to know when you are looking at enzyme is that they are catalysts. So the definition of enzyme also is what? The characteristics. So we are going to be saying they are enzymes. They are catalysts. Yeah, and I explained the meaning of catalyst where I said a catalyst is something that speeds up a chemical reaction. Then the next characteristics which you need to know under enzyme is that they catalyze both forward and backward reaction. They catalyze both forward and backward reaction. So what do we mean when we say they catalyze both forward and backward reaction? So meaning some catalysts can be used to break down substances, while others can be used to build back the what? The substance. So this is the third characteristics of enzyme. Now we are going to look at the fourth characteristics of enzyme, which is enzymes are specific in nature. So what do you mean when you say enzymes are specific in nature? So when you say that enzymes are specific in nature, remember what I said when we talked about the lock and key mechanism. We said that only one substrate can be worked on by an enzyme because they have what? A complementary shape, meaning only one substrate can fit into one enzyme, just as a key fits into one specific door. So that's where you need to understand. Then, after looking at the characteristics, we are going to discuss now the factors which affect the rate of enzyme. So this is where you need to pay attention, because this part is the part which they like bringing in questions, like the question which we see. Yeah. So what are some of the factors which can affect the rate at which enzyme work? So we have factors. So the first factor which you need to know is temperature. So how can temperature affect the way enzyme work? So temperature affects the rate of enzyme in the sense that when you increase the temperature, you increase the what? The rate at which enzyme work until you reach a specific temperature known as the optimum temperature. So at optimum temperature, that's where an enzyme works the best. So what do we mean? So whenever you are increasing the heat, for example, you are increasing the activity of an enzyme until you reach a certain point which is known as the optimum temperature. So most question on temperature will come in form of a graph. So most question comes like this. Then on the x-axis you have temperature. Then here we have the rate. So whenever you see a graph looking like this, it means we are talking about a graph of temperature. We said that the first thing is if you increase, so increase in temperature increases the rate of enzyme. Yeah, so that's the first point you need to understand on temperature. So whenever you increase the temperature, you increase the rate of enzyme. Then the second thing we say that enzyme work the best at a certain temperature, which is known as the optimum optimum temperature. So in case they ask you to label on the diagram where the optimum temperature is, this is where the optimum temperature is. Yeah. And remember, whenever you are looking at diagrams on enzyme, mostly they are the same when you are labeling them. So the next thing we say that at optimum temperature, enzyme works the best. Yeah. Then there is also another point which you need to understand on temperature. So the third thing you need to understand on temperature is that after you pass the optimum temperature, the enzyme will become what? Denatured. So after the optimum temperature, the enzyme, so after optimum temperature, enzyme becomes denatured. So what do you mean when we say denatured? So denatured, this is the disturbance in the shape of the enzyme so that the enzyme and the substrate won't bind together. So whenever you hear the word denature, just remember that this is the disturbance in the shape of the enzyme and the active site, so that to prevent the substrate from binding. Yeah. So this was our first factor which affects the rate at which enzymes work. The next factor is known as pH. So here again is where I need to pay particular attention, because the graph of temperature and pH are slightly similar in some case, but they are different. When you talk about the graph of temperature, we are saying that when you increase the temperature, you increase the what? The rate of enzyme. But when we talk about the pH, we are determining whether the enzyme is acidic or basic. We know that the stomach is acidic, and we also know that the small intestine are basic. So this is where now you have to determine which enzymes work best in the stomach or the duodenum using the pH. So we said that our second factor is pH. So when we are talking about pH, we are saying that this is the measure of how something is acidic or basic. Yeah. So how does the pH affect the enzyme? So before we talk about how the pH affects enzyme, you need to understand first what is known as the pH scale. So what is a pH scale? So a pH scale is a scale we use to determine whether something is acidic or basic. Yeah. So mostly the pH scale will be brought in form of a diagram. So you have pH here, then you have a rate on the Y, then on the X we have pH. Yeah. So like I said, the graph of pH and the graph of temperature, they are the same. The only difference is this one is measuring what? The acidity and the basic of a solution, whilst the temperature measures the what? The rate at which the reaction is moving. So again, we have the pH, and the pH also have a pH which is known as the optimum pH. Yeah. Here the optimum pH, we are saying that this is the pH at which enzyme works the best. So meaning in our case, here you have to decide whether an enzyme works best at acidic or it works best at alkaline. An example here we can have is the digestive system. We have the stomach, which is acidic. So in the stomach, enzyme which will work best are enzyme which works under acidic condition. And in the duodenum, enzyme that work the best are enzyme which works under basic condition. Yeah. So now what you need to understand, so we have, so the pH scale ranges from 1 to 14, and we have the mid, which is 7. So from 1 to 6, this is acidic. Then from 8 to 14, we have basic. So these are the two things which you need to know under the pH. And we say that the optimum pH here is depending on the site where the enzyme is. If it is in the stomach, it will work best under acidic condition. Then 7 is neutral. Yeah, so these are the two main factors which you need to understand in detail on enzymes. Then we have the other factors like enzyme concentration, we have substrate concentration, so what is the meaning of these two and how do they affect the rate at which an enzyme works? So when we talk about the enzyme concentration, we are talking about the amount of enzyme. So if we increase the amount of enzyme, of course the rate of the reaction will increase because you are increasing the enzyme. So when you increase the enzyme concentration, you increase what? The rate. Then again, if you increase the substrate concentration, you increase what? The rate. So on enzyme concentration and substrate concentration, these are the things which you need to know. Whenever you increase the enzyme concentration, you increase the rate. Then another factor which you need to pay particular attention is inhibitors. So inhibitors are substances which slows or completely stops what? The activity of enzymes. So we have a lot of inhibitors. So here what you need to know that all poisons are what? Are inhibitors. So we are saying we have inhibitors. So inhibitors is a substance that slows or completely stops the rate of, the enzyme activity. The rate of enzyme. Then you need to understand that we have two types of inhibitors. This is the point which you need to pay attention. We have one which are known as competitive and we have non-competitive. So when you talk about competitive and non-competitive inhibitors, what should come into your mind is the enzyme itself. So here these inhibitors work on the site. We have two sites, I told you. We have an active site and we also have an allosteric site. So competitive inhibitors, they bind to the active site. So we know what happens when an inhibitor binds to the active site, meaning it will block the substrate from binding where? To the enzyme. Then when we talk about non-competitive enzyme, these are enzymes that binds to the allosteric site. Yeah, so what happens when an inhibitor binds to the allosteric site? So it changes the shape of an enzyme. We say that once the shape of an enzyme changes, meaning the substrate, which is the key, won't fit because the shape has changed. So these are the top important factors which you need to know when it comes to enzymes. So the final part on naming enzymes, you can go and study on yourself. So concluding today's lesson, we are going to be revising now this question which came on enzyme. So the question is saying figure 2.2 is a graph showing the effect of pH on the rate of enzyme controlled reaction N and O. Yeah, so this graph is showing us the graph of what? Of pH, which I'm from explaining. So the first question is saying what term is given to the parts labeled X and Y. So remember I told you that enzymes, same as temperature, have a specific point which is known as the optimum pH. So meaning X and Y are known as the optimum pH. So this is how simple biology is. Then the second question is saying which reaction is under acidic condition. So remember I told you that the pH is divided into two. We have 1 to 6, which is acidic, 8 to 14, which is basic. So in our case, if you look at X and Y, you will see that the optimum. So whenever you are looking for an enzyme under acidic condition, we follow what is known as the optimum pH, because that's where the enzyme works the best. So meaning if this enzyme is below 7, meaning it works the best under acidic condition. Then if it is above 7, meaning it works best under alkaline condition. So if you can look at the diagram, we have X, which is at 4, and we have Y, which is at 11. So meaning X is below what? 7. So meaning X is under what? Acidic condition. So meaning the graph of N is under what? Acidic condition. Then the third question is saying which reaction could only be for a protein food? Give a reason. So the next question they're asking us, which reaction could be for a protein food? Give a reason. So we know that digestion, so whenever they ask you this question, think about digestion. Where does the digestion of protein takes place? So mostly the digestion of proteins takes place in the stomach. So remember, if the digestion of proteins happens in the stomach and the pH in the stomach is acidic, which means the reaction which will be for a protein is what? N, which works best under acidic condition. So here is a reaction N, and the reason is that the optimum pH is acidic, as you can see on the diagram. So that's how you can explain. Then the final question is saying explain the law of enzyme in metabolism. So metabolism, this is what? The forming of what? Of substance. So whenever a substance is what? This is the breakdown of substances. So for example, when we have carbohydrates, when you eat, of course those carbohydrates need to be broken down. So that process of metabolism is what they are asking here. What is the law of enzyme? So we say that enzyme what speeds up the metabolism. So that is the role of enzyme in metabolism. Their function is just to speed up the reaction. Then coming on to our second and final question, which is saying the figure below shows in order 4 stages in which an enzyme controlled chemical reaction may occur. The first question is saying identify M, N, O, and P. So in the previous video, I talked about M, which is the substrate. I also talked about N, which was the active site, where the substrate will bind. Then we talked about O, which was the allosteric site, which is just the other part of the enzyme. Then when we talk about P, we say that after an enzyme works on the substrate, it will form what are known as products. Yeah. Then part B is saying, by referring to the diagram, explain why only small amount of enzyme is needed to catalyze a reaction involving many molecules. So here you can explain the lock and key mechanism, where we say that each enzyme has a specific substrate or a narrow range of similar substrate which it can catalyze because of the complementary shapes. So here you can say because the enzyme has a complementary shape, then the final question, which is saying at high temperatures enzymes are denatured and can no longer act as catalyst. By using letter M and N, suggest what happens when an enzyme is denatured. So here I told you that when an enzyme is denatured, it loses its function. And what happens? The first thing is M, which is the substrate, and N, which is the active site. So when we're talking about the substrate and the active site after the enzyme is being denatured, it means that the enzyme or the active site will be deformed. So once the active site is deformed, the substrate won't bind to the active site. Therefore, the enzyme can no longer do or perform its function. So this is where the topic of enzyme ends. Be sure to subscribe, comment, and share if the video was helpful.